Welcome to section 13 of metabolism. In this section, we'll be discussing ethanol metabolism. Let's get started. This is the metabolic map provided in section 1 of metabolism. In this video, we're focusing on ethanol metabolism. I haven't included ethanol metabolism in this figure because it's a relatively short pathway and can make this bigger map look pretty messy, but ethanol normally feeds into acetyl-CoA right here. Let's zoom up on the detailed figure of ethanol metabolism so you can see what I'm talking about. This is a detailed figure of ethanol metabolism, which can be found in section 13 of metabolism. Notice that ethanol is converted into acetaldehyde, then acetate, and finally acetyl-CoA, where it can be used in the TCA cycle or for the synthesis of ketones. You can see from the figure that alcohol dehydrogenase right here and acetaldehyde dehydrogenase right here are both enzymes necessary for the metabolism of ethanol. Also notice that these enzymes are responsible for the production of NADH. Accumulation of NADH is responsible for many of the findings seen in alcoholism. In this figure, you can see ketone bodies, lactate, and triglycerides. Alcoholism results in derangements of these three compounds, and these represent the high-yield takeaway points from ethanol metabolism, which we're going to discuss now. From prior videos, you should know that NAD and NADH are necessary for several biochemical reactions. In the TCA cycle, recall that malate is converted to oxaloacetate and produces NADH in the process. In alcoholism, NADH levels are high, and this inhibits this reaction, resulting in the depletion of oxaloacetate. Recall that oxaloacetate is required for gluconeogenesis, so inhibition of this step is responsible for impaired gluconeogenesis. We can deduce then that alcoholism can cause fasting hypoglycemia. From the figure, you can see that ATP and NADH also inhibit other enzymes in the TCA cycle. Therefore, alcoholism impairs the TCA cycle and results in the accumulation of acetyl-CoA. If acetyl-CoA is unable to be used in the TCA cycle, it's shunted towards the synthesis of ketone bodies. This explains why alcoholic patients can develop a ketoacidosis. Next, notice that the conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate causes NAD to become NADH. This depletes the cell of NAD, and glycolysis cannot continue to occur unless NAD is regenerated. Recall that under normal conditions, the cell replenishes NAD through the electron transport chain. In alcoholism, however, NADH levels are high, and this overwhelms the electron transport chain. Additionally, the high NADH inhibits the TCA cycle. This causes acetyl-CoA levels to rise, and pyruvate can no longer be converted into acetyl-CoA. Instead, the pyruvate is shunted towards lactate. Notice that the conversion of pyruvate to lactate regenerates NAD. This allows the cell to continue glycolysis. The increased production of lactate is responsible for the lactic acidosis seen in alcoholism. Finally, notice that the conversion of glycerol 3-phosphate to DHAP produces NADH. In alcoholism, NADH levels are high, which inhibits this step. This means triglycerides within the liver are unable to be properly metabolized. This results in an accumulation of fat in the liver and is responsible for fatty liver disease, which is commonly seen in alcoholics. Okay, let's do a question. A 65-year-old male presents to the clinic for a routine evaluation. Physical exam is significant for mild hepatomegaly. Labs reveal an AST of 230 and an ALT of 115. This patient's physical exam and lab findings are most likely due to impaired gluconeogenesis, resulting in an accumulation of what metabolite in hepatocytes. From the question stem, we know that this patient has AST and ALT elevations. Recall that these are elevated in patients with liver damage, and an AST to ALT ratio of 2:1 is suggestive of alcoholic-related liver damage. In this case, the patient has an AST of 230 and an ALT of 115. This is an exact 2:1 ratio. From the figure, we can see that triglycerides are normally metabolized into glycerol and fatty acids. The glycerol can be converted into glycerol 3-phosphate, which can then be converted into DHAP and produces NADH in the process. In alcoholism, however, NADH levels are high, which inhibits the conversion of glycerol 3-phosphate to DHAP. This means glycerol and triglycerides within the liver are unable to be properly metabolized. This results in an accumulation of fat in the liver and is responsible for fatty liver disease. So in answer to the question, this patient's hepatomegaly and elevated transaminases are likely due to impaired glycerol metabolism in the gluconeogenic pathway, resulting in an accumulation of triglycerides in hepatocytes. Okay, there are several more points to be familiar with. Fomepizole inhibits alcohol dehydrogenase. This is important because it can be used to treat patients with methanol or ethylene glycol intoxication. Methanol and ethylene glycol have a similar structure to ethanol, so it should make sense that they're metabolized by alcohol dehydrogenase. Fomepizole inhibits the metabolism of the toxic metabolites of methanol and ethylene glycol, so it can be used to treat this type of intoxication. Notice from the figure that disulfiram inhibits acetaldehyde dehydrogenase. This can be used to deter alcoholics from consuming alcohol because it results in the accumulation of acetaldehyde. Acetaldehyde can cause uncomfortable side effects such as sweating, nausea, vomiting, and flushing. There's also a genetic condition that results in a deficiency of acetaldehyde dehydrogenase. It's most common in Asian populations and results in similar symptoms as a patient who has been given disulfiram. One final point before we wrap up with another question. Alcoholism can cause thiamine deficiency due to malnutrition and malabsorption. This is perhaps the most important point to be familiar with regarding alcoholism. Recall that thiamine, or vitamin B1, is an important cofactor for several enzymes. Some of these include pyruvate dehydrogenase, alpha-ketoglutarate dehydrogenase, transketolase, and branched-chain ketoacid dehydrogenase. Okay, let's do another question. A 41-year-old homeless male is brought to the ED after being found unconscious in the street. The individual who brought him to the hospital stated that he smelled of alcohol. After proper resuscitation, it is determined that he has a vitamin deficiency resulting in memory problems. What enzyme involved in the synthesis of DNA is likely impaired as a result of this patient's vitamin deficiency? Hopefully from the question stem you notice that this patient has chronic alcohol use resulting in memory problems. Recall that chronic alcohol use can result in thiamine deficiency. As we just learned, thiamine deficiency results in impairment of several enzymes, including transketolase, which is involved in DNA synthesis. Recall from the section on the HMP shunt that transketolase is required for the conversion of fructose 6-phosphate to ribose 5-phosphate. Ribose 5-phosphate can then be used in nucleotide synthesis for DNA. Notice from the figure that transketolase requires thiamine as a cofactor.